We're here with Lucie Delemotte from the Royal Institute of Technology in Stockholm, Sweden. She's giving a talk on Tuesday entitled The Details of G-Protein Coupled Receptor Activation via Data-Driven Molecular Modeling. Welcome, Lucie.
Thank you so much for having me.
Yes. Would you mind talking briefly about some of the more recent exciting developments in structural biology that have allowed researchers to understand the cellular membrane in greater detail?
So it's really interesting because in 2013 we had this so-called resolution revolution going on in cryo-electron microscopy, and we went from having a structure of a specific membrane protein to having structures of many of those. So now we can take an approach where we actually compare these different membrane proteins and see how they work in relation to one another. So we used to think, everything just behaves like one protein, but in fact, with this resolution revolution, now we see that it's actually not the case and that different family members behave quite differently.
And tell me what part computer modeling is playing in better understanding this complex structure.
Yeah, sure. So that's really interesting because my lab actually specializes in using so-called molecular dynamic simulations. So that's a technique that uses the methods from physics to model how the membrane protein is evolving over time. So instead of having like a structural snapshot like we have from experimental methods like cryo-electron microscopy, we end up with a movie of how the protein is working over time. And so we recycle methods from machine learning, artificial intelligence, and just adapt it to our needs.
Please elaborate on some of the more exciting discoveries you've made.
Yeah, so what I'll be talking about, for example, in the talk tomorrow is these G-protein coupled receptors. The way they work is there's a ligand that comes from the outside of the cell, it binds to the receptor, and that triggers a conformational change in the receptor that then has an effect downstream in the cell. And so we have realized that we can sort of describe the protein in sort of a layered structure, where the layer that is influenced by ligand binding will then influence the layer that's right below, which in turn will influence the layer that's right below until we have this this so-called allosteric change at the bottom of the receptor. So you can think of it as like a domino effect almost. But instead of being really like a domino where like one domino flips the next domino over, it's like a probabilistic domino effect.
And so, tell me just a little bit as well. It sounds like you work with quite a variety of fields. You were mentioning that you're borrowing from artificial intelligence and other. Tell me a little bit about that.
Yeah, so that's interesting actually. So I'm a chemist originally, but I went to universities to study biology. I really liked biology, and then I realized I didn't like the way biology was taught. I really wanted to apply the methods from chemistry and physics that were more quantitative, and I think it doesn't make sense anymore actually to divide science in like these disciplines because they actually kind of work together. And why not use a method from a different field? Like why, if it's going to help your project, I don't see why.
That's right. Yeah. Bring everybody together.
Yeah, that's right.
Well, thank you so much for coming and talking with us today.
Thank you. Thank you for having me.